Hi there, my name is Brandon Shellstrom, and today I'll be doing a more detailed overview of how to extract brain skull samples from the porcine skull. So to start off with, I'll be going over the soft tissue removal, which still be glossing over rather quickly since it's mainly done to reduce clogging of the bone saw blade for later steps. Soft tissue really tends to clog up the blade teeth of the bone saw, which takes a significant amount of time to clear. Since we know that the brain tissue increases in orders of magnitude of stiffness post-mortem, we want to extract these samples as quickly as possible. So therefore, removing as much soft tissue as possible to reduce that clogging is ideal. So the main purpose of this is to expose the frontal and occipital bones through a series of cuts that remove a lot of the soft tissue. More of the soft tissue, additionally to the soft tissue, will be the muscles controlling the eye region, directly lateral to the parietal bones. And then finally, a lot of soft tissue at the posterior end of the occipital bone. Next, this is a little bit where user choice comes in, as the user must create a horizontal cut 10 to 15 millimeters from the most superior portion of the occipital bone. The more bone that is cut away, the faster the dissection process, but the higher risk of actually causing damage to the delicate interfaces at the brain. However, we can see that regardless of the direct cut depth, removing a lot of the lateral and posterior tissues, we can see this nice brain capsule supported right in the middle here. And as a good validation check from this direct sample, if we can look here, we see these thin, delicate, mineralized structures that are found within the sinuses that are still intact, even though a lot of the surrounding brain region was removed, which we're then claiming means that the delicate interfaces inside the actual cortex are still going to be intact as well. Here's a little point of interest to bring up as the difference between age and breed. This nice porous structure that we have on the right only occurs in mature pigs of very specific breeds, as we have a sinus region that begins just anterior to the frontal bone and grows posteriorly and superiorly over time, depending on the age and breed of the pig. Which changes the overall functional thickness we're drilling through. So a lot of these immature pigs, this highly trabeculated bone doesn't occur yet. So it's simply a lot higher functional thickness to get through to get samples, which simply must be ground away slowly to avoid damaging this delicate brain region. Therefore, once the cortex has been exposed, we're going to create a series of burr holes using a craniotome drill bit. It's a special drill bit that allows for drilling only up until the softer dura mater underneath has been punctured. So we can see this drill bit here that once the drill bit punctures the skull, it won't be allowed to spin. And this is done through two separate interlocking drill bits. So when the inner drill bit is pushed into the outer drill bit, they're both allowed to spin. However, once it punctures through the skull, it cuts off and no longer allows either drill bit to spin. In standard craniotomy, the next step would then be to peel the dura mater off the inner cranium. However, this is where we're going to differ a little bit. We can see here this nice little structure inside the actual skull that we have laid out on the left. Next, we simply connect the two inline cuts, the set of parallel orthogonal cuts, where we're claiming the dura mater in between these two samples should still be intact. And this is done using a bunch of very light passes. So the way this bone saw motor came from is that the sample is generally slightly pressed into the skull using a set of weights, which then just slowly cuts through the skull. I'm using this more of an inverse fashion, where I use the weights of the actual blade to be pressed into the skull. We can see kind of the results of that here, where we have the same sample as the previous slide up top, and then a different previous sample where we didn't have the burr holes, showing that once we remove the skull, we have our nice dura mater underneath. Next, we're going to remove the dura mater. We're just simply tracing the previous cuts with a scalpel. And here, I think this introduces an issue with these samples quite well. As we can see, this sample has been fully separated from the skull, and it's very easy and soft to move around. It actually looks like it's detached. However, this isn't the case. And this is mainly because this sample is dealing with the dura mater mechanics. So the trick that I found was leaving the most medial edge of the sample of the skull tissue still attached, and then cutting away the dura on the other sides of the sample, since it allowed for a much easier cut, since we're still dealing with the mechanics of the skull. However, once the skull has been cut away, now the only thing supporting this upper skull piece is this below soft tissue. So now we're dealing with the pure cortex mechanics, which makes cutting this dura significantly harder. However, since we have the other three sides of the dura cut, this becomes a lot easier of a cut to do. And finally, the last step, the step that I'm still working the most on is actually extracting the sample from the brain. This is done by inserting a scalpel through the perpendicular edge and pressing on the bottom of the sample with the sample inverted. This is because if you simply grab the top of the skull with a set of forceps, the adhesive forces of the cortex and of the underlying arachnoid layers are so significantly stronger compared to the strength of the interface between the arachnoid and dural barrier, which often tends to separate if you just simply pull up on the sample. However, if you turn the entire head upside down, or allowing the force of gravity combined with pushing on the bottom of the sample with a scalpel is enough to overcome its adhesive forces to actually remove the sample without damaging that interface. And here's kind of nice little validate. Here's a nice little check of some of the samples we've removed from the porcine skull. So up top, we can see the nice white and gray matter from our cortex, with our dura matter as this pink little region between them, and finally our skull below that. Some of our later samples, we can even see the arachnoid showing up, which we have, if we compare the cortex on the left here to the cortex above, we can see this is a lot darker since our arachnoid is actually present. And even here, we can see our arachnoids present in a red shape. And even if we take this middle sample and separate it apart, we have our skull and our dura and our arachnoid is very clearly this red substance to the side of it, which can clearly be supported and is still connected to the cortex. And then if we make little cuts along the inside of this arachnoid, we can see the P matter would be found just below that. Again, it is only a single cell thick. So while we're present, we're sure it is there because this arachnoid is still connected to the cortex. It's still very difficult to see without a microscope. And then finally, in this bottom left image down here, we can see a sample that's even inverted with our skull superior to the cortex. We can see that the cortex is still attached. It doesn't want to fall away immediately, even however delicate this interface is. So thank you for listening, and I hope you enjoyed the talk here.